Hello students, welcome to BioDigest. In this particular video, we will discuss about cell division and the types of cell division. So we will see what is cell division, the definition. How will you define cell division? Cell division is the process by which a single cell will divide into two or more daughter cells with the same genetic constitution. So here I am showing a single cell with a nucleus. This is the nucleus. This will divide into two daughter cells or maybe more in some cases with the same genetic constitution. That is what is known as cell division. Now, what is the need for cell division? Why are the cells dividing? So, the reasons are, first is cell division is actually taking place for growth and development. Second is, it is actually helping in the repair of damaged tissues. That means if you are having an injury, so it will be helping in repairing that. That is why you might have experienced when you are having a small injury in your hand or in any part of the body. After 1 week, you can see that the injured portion, it will come back to normal. So actually what is taking place? The repair of the damaged cells is brought about. How? It is actually brought about with the help of cell division. Then another is, it is helping in reproduction process. You might have heard about asexual reproduction. Okay, asexual reproduction means without the involvement of sex organs. So in this asexual reproduction, actually cell division is playing a very important role. So all these are the reasons why a cell is dividing. Now we will see what are the types of cell division. Basically there are two kinds of cell division. Which are they? It is mitosis and meiosis. What is mitosis? Mitosis is the type of cell division that will be resulting in growth and development. Now a question can come for the board exam. Where does mitosis and meiosis takes place? So where does mitosis happen? Mitosis will take place in the somatic cells of the body. That means normal body cells. Now what is meiosis? Meiosis is the type of cell division that will be resulting in the production of gametes. What is the meaning of gamete? You might have heard about sperms and egg. Okay, I am speaking about human beings. Sperms in the case of males and the egg in the case of females. So meiosis is the type of cell division that is actually resulting in the production of gametes. So where does meiosis take place? Meiosis will take place in the reproductive cells. Now, we will learn in detail about mitosis. But before the cell enters into mitosis, the cell will undergo a period of rest. That is what is known as interphase. So this is actually the resting period. or resting phase of the cell. So in this stage actually, no visible change will happen to the chromosome as such, but active synthesis of DNA will be taking place. That means this is the resting period during which the cell is preparing itself for division. That resting phase is what is called as interphase. So the appearance of the cell will be like this. I am showing an animal cell. Okay, so this is the cell membrane. Animal cell usually has only cell membrane. This is the nucleus. Nucleolus will be there. And the chromosomes, I told you, chromosomes as such we cannot call it as chromosomes. Instead it will be appearing in the form of thread-like structures known as chromatin threads. Okay, so this will be the appearance of a cell during interphase stage. And of course, there will be centrosome containing centrioles present in the case of an animal cell. Question can come for the board exam. Name the type of cell that is shown in the diagram. So if this is the type of the cell that is shown in the diagram, you can say that it is an animal cell. So another question will be there. Give reason to support your answer. So what are the reasons? Why are you saying that this is an animal cell? So the answer is very simple. You can write that the cell is having only cell membrane. Then you can write centrosome containing centrioles are present. So these two are the identifying features for an animal cell. Now what about a plant cell? The same thing I will show it in a plant cell. So if it is a plant cell, what will happen? The plant cell will be having cell wall as well as cell membrane. So the outer one is cell wall. The inner membrane is cell membrane. Then nucleus of course will be there, nucleolus and chromatin threads. Now another thing is that centrosome containing centrioles will be absent in the case of a plant cell. And cell wall will be present. If these two things are there, you can identify the particular cell as a plant cell. Okay? So that is about interphase, that is the resting phase of the cell. Now before going into mitosis, let me just give you a brief idea with regards to meiosis. Because meiosis is not there actually in the syllabus, only a brief idea is to be given. We are not learning in detail about meiosis. So we will see meiosis. Meiosis is known as reduction division. Again, this is an important question. Why meiosis is called as reduction division? It is because during the time of formation of gametes, the chromosome number is getting reduced to half. That means, in the case of human, I told you sperm and egg will be there. So in the case of sperms, there will be 23 chromosomes. So humans are having 23 pairs of chromosomes. Okay? Or we are having 46 chromosomes. So 23 out of the 23 pairs, 23 will be present in the sperm and 23 will be present in the egg. So during fertilization, what will happen? During fertilization, the fusion of sperm and egg will be taking place. Okay? So as a result of which zygote will be produced. So when the zygote is produced, it will be having 23 chromosomes of the sperms, 23 chromosomes of the egg. Thereby the normal diploid number is reacquired. This is the spelling. Diploid number is reacquired. So diploid number is represented as 2n. So this 23, you can call this number as haploid number, which is represented as n. Okay, so haploid, that is n. 23 chromosomes are there in the sperms. Again 23 chromosomes are there in the egg. So when a fertilization happens, the 23 chromosomes of the sperms will fuse with the 23 chromosomes of the egg. Thereby the normal diploid number is reacquired. So that is why meiosis is called as reduction division. So this is an important question that will come for the board exam. Now moving to mitosis. So mitosis is the cell division, I told you, that is actually resulting in growth and development of the body. So mitosis, it is divided into two stages or two phases. Karyokinesis and cytokinesis. What are these actually? Karyokinesis. Karyo means something related with the nucleus. So what is karyokinesis? There will be some changes taking place to the nucleus at the time of mitosis. All those changes finally will result in the division of nucleus. So that is what is known as karyokinesis. So how will you define karyokinesis? Karyokinesis can be defined like this. The nuclear changes which finally leads to the division of nucleus is called as karyokinesis. I repeat, the nuclear changes which finally result in the division of nucleus. So what is cytokinesis then? Cytokinesis is something related with cytoplasm. So what is cytokinesis? Cytokinesis, in simple terms, it is the division of cytoplasm. So this is the division of cytoplasm. And this is the division of nucleus. Okay, so karyokinesis, it is the nuclear changes which finally result in the division of nucleus. Cytokinesis is the division of cytoplasm. So karyokinesis, again it is divided into 4 stages. What are they? Prophase, Metaphase, Anaphase and Telo Phase. So when the nucleus is undergoing prophase, some changes will be there that will be happening to the nucleus. Again, when it enters into metaphase, some other changes will be noticed. Then anaphase means another changes, and telophase means another changes, and finally it will result in the division of nucleus into 2. So we will be seeing that in detail in this particular video. This series of steps that is taking place in karyokinesis, you can remember like this. PMAT. P is prophase, M is metaphase, A is anaphase and T is telophase. So I told you before the cell enters into karyokinesis process, the cell will undergo a period of rest, that is what is known as interphase. We will discuss about interphase in the next video. So coming to prophase. So this is over. Next is prophase. What will be the changes that will be happening during prophase? So again I am showing the animal cell. Prophase, let me tell you, it is divided into early prophase and late prophase. So I will draw the diagram, both the diagrams here. So previous diagram I told you centrosome containing centrioles were present. So here centrioles will be formed like this. Okay, so first is this. This is actually, you can mark it as centriole. Then we are bothered about the nucleus. This is the nuclear membrane. Labeling is very, very important. You need to show minimum 4 labelings. Nuclear membrane. That means nuclear membrane is present. This is nucleolus. Nucleolus is present. I am speaking about early prophase. Okay, this is early prophase. Now, you will be able to see chromosomes now. Previously, it was in the form of thread-like structures. Now, you will be able to see chromosomes like this. Chromosomes will become distinct. Okay? So, all these changes are taking place during early prophase. So, what are they? In early prophase, you will be able to see the centrioles. Then, nuclear membrane is clear. It is present. Nucleolus is present. Now, the chromosomes are actually visible. So, all these are the changes taking place during the early prophase. Now, the same diagram, I am going to make some changes so that it will become late prophase. Okay, so this is the diagram for the early prophase. You just see centrioles are here, nuclear membrane is there, nucleolus is there, chromosomes are scattered. Now, what are the changes that will happen to this cell when it enters into the late prophase stage? So, this centrioles, this will move to 2 opposite poles. So, this diagram itself I am going to make the changes. Okay. So, this is the centriole. Here also you can show one. It's moving to 2 opposite poles. So this is known as the aster ray. Aster rays. Okay. Now I told you nuclear membrane is present during early prophase. The nuclear membrane will disappear. So nuclear membrane will not be there. So we can rub this. Nucleolus also will disappear. So nucleolus is also not there. The condition of the cell will be like this. So this is already marked aster rays. You can mark this as centriole. Then one major important thing will happen. You will be able to see one spindle fiber that will be holding the chromosomes like this, which will run from one end to the other end. Okay? Like this. So this is known as spindle fiber. So spindle fibers will be appearing. And the spindle fibers will be getting attached to the chromosomes like this. Okay, if this is the chromosome, the spindle fiber will be getting attached to the centromere of the chromosome. So that will happen during the late prophase. So what are the changes during the late prophase? nuclear membrane will disappear, nucleolus will disappear, centrioles will move to two opposite poles, then spindle fibers will appear and it will be running from one pole to another pole, and in between they will be having the chromosomes. So that is about the late prophase. So prophase as a whole, what all changes are taking place? Early prophase, you know, the chromosomes are distinct and visible. Nucleolus, nuclear membrane is there. Centrosome containing centrioles are present. But when it enters into the late prophase, nuclear membrane disappears, nucleolus disappears, spindle fibers will appear in between the two opposite poles. So all these are the changes that is taking place during late prophase. Moving to the next phase, now we have completed interphase, that is the resting stage, prophase. We have completed early prophase and late prophase. Next is metaphase. So, what will be the appearance of a cell during metaphase stage? This is the cell. Now, no nuclear membrane and all, okay? So, just we have to show that aster on two opposite poles like this. Now, meta, that means something related to equator. So here what happens is that just at the center I am drawing just a slight light line like this. All the chromosomes will be lined up at the equator in a single plane like this. After which we will rub that line, okay? So I am showing 4 chromosomes. All these lines I am rubbing. So you can now see it's aligned in a single plane at the equator. So that is the most important feature of the cell during metaphase stage. Now what happens? I told you spindle fibers will be there. It will be holding the chromosome by the centromere region. So this is the centromere. It will be holding the chromosome by the centromere region. So this is the appearance of the cell during metaphase stage. So you can label the diagrams. I told you this is chromatid of the chromosome. Centromere you can label. This is the spindle fiber. Then this is aster ray. Okay. So this is this appearance of a cell during metaphase stage. So what are the features? The chromosomes are lined up in a single plane at the equator. Second is the chromosomes are actually held by the spindle fibers, or the spindle fibers will be getting attached to the centromere of the chromosome. Okay, you can see that the spindle fiber is getting attached to the centromere of the chromosome. So all these are the features of the cell during metaphase stage. Now moving to the next stage, that is anaphase. What will be the appearance of the cell during the anaphase stage? So this is the cell. Aster. In anaphase, again this can be divided into two: early anaphase and late anaphase. What is actually happening during early anaphase? You know the structure of a chromosome. The chromosome is like this. Now in the metaphase stage, you know that there is spindle fiber that is getting attached to the centromere of the chromosome like this. So what happens is that when it enters into the early anaphase stage, the spindle fibers holding the centromere will begin to shorten or constrict. Constriction of the spindle fibers will take place, as a result of which the chromosome will split into two. So this is what is known as chromatid. Okay, it will split into chromatids, two chromatids, and it will slowly move to the two opposite poles. This is actually taking place due to the shortening of the spindle fiber. So in early anaphase stage, you can show like this. The centromere will be almost towards the center. Okay, I'm showing that like this. 4 chromatids. Here also same thing. When you are drawing, please be very careful. Centromere should be facing the pole. So shortening of spindle fibers will be taking place. It's actually like this. I'm showing the early anaphase stage. Okay. Now, when it enters into the late anaphase, what you can see? The chromatids will be reaching the poles during the late anaphase, like this. In addition to that, a furrow will be appearing in the middle of the cell like this. So these are the changes during anaphase of the cell. So what all things are happening? During the anaphase stage, the spindle fibers are actually getting constricted. As a result of which the chromosomes will split into two chromatids. This is the spelling. And the two chromatids will be moving to the two opposite poles. When you are drawing the diagram, please be very careful. Centromere should be facing the polar region. Okay, poles, centromere. This is the chromatid. Labeling should be there. And towards the end, that is late anaphase stage, the chromatids will reach the two opposite poles. You can see the appearance of a furrow in the middle of the cell. A furrow will be appearing in the middle of the cell. So all these are the changes that is taking place during anaphase stage. Now comes the last phase, that is Telo Phase. Now in Telo Phase, what is actually happening? The chromosomes will be reaching the two opposite poles. I told you there is a furrow, so we have to show that furrow also here like this. Furrow is actually deepening now. Okay, so this chromosomes that have reached the two ends, it will thin out in the form of chromatin fibers. So 4 chromatids were there. Okay, so this will thin out in the form of chromatin fibers. Spindle fibers will be disappearing. Now what happens is that you can see the formation of nuclear membrane. Nuclear membrane will reappear. Nucleolus will reappear. This is the chromatin fibers. It was actually in the form of chromosomes. It will thin out and it loosens to form the chromatin fibers. So this is the nuclear membrane. Nuclear membrane reappears. Whatever was happening during prophase, just the opposite will happen in telophase. Okay, this was disappearing in prophase. So that will again reappear in telophase. Now nucleolus also reappears. Now what happens? This furrow, this will be deepening. Now you can see that the nucleus is actually divided into two. Okay, there was only one nucleus when the cell division, that actually the nuclear division began. So when the cell reaches telophase stage, you can see that the single nucleus that was present, it will divide into two nuclei. Now what happens is that the furrow that was there, it will deepen further. Okay, like this. Resulting in the division of cytoplasm. Now the cytoplasm is also dividing, as a result of which we are actually getting two daughter cells. So a single cell was there, now it will divide and we are getting two daughter cells. Okay, of course there will be the presence of centrosome and all. I am rubbing this nuclear membrane. So centrosome will be there. You have to show it on both the sides. So this will be the appearance of the cell when it undergoes telophase stage. Now another question can come. What is the difference between mitosis taking place in a plant cell and an animal cell? So what are the differences? Now you can recall and you can just identify that differences. What are the differences? I already told you. One is, you can write, in the case of animal cell only cell membrane is present. In the case of plant cell both cell wall and cell membrane are present. Then related to mitosis, what are the differences? Centrosome containing centriole is present only in the case of animal cell. Centrosome containing centriole is totally absent in the case of a plant cell. Then another is, here I told you it is by furrow formation. Okay, this is known as furrow. Animal cell, the division of cytoplasm is taking place by furrow formation. In a plant cell, the division of cytoplasm is taking place by cell plate formation. It is known as cell plate. A plate will be formed like this, which is known as cell plate. So the division of cytoplasm is taking place by cell plate formation. Okay. So in animal cell, it is by furrowing of the cytoplasm. The cytoplasm will divide into two. But in the case of a plant cell, cell plate formation will be taking place, which will be resulting in the division of cytoplasm. So I hope the differences between plant cell and animal cell with regards to mitosis is clear to everyone. Okay, then I explained about meiosis also. That is actually called as reduction division. All those reasons have been explained. Now what is the significance of mitosis? How will you write the answer? Mitosis, you can write it will help in the growth and development of an organism. The most important point is it will help in maintaining the same chromosome number. Sometimes name the following question can come. Name the type of cell division that is associated with maintaining the same chromosome number. So what is the answer? It is mitosis. Whereas meiosis, I told you, it is actually related with the production of gametes. It is the type of cell division that leads to the production of gametes. So that is the most important thing with regards to meiosis. Then all the details are not there in the syllabus, but I will just give a brief idea about meiosis. In meiosis, what happens is that variations can happen because crossing over will be taking place between the maternal chromosome and the paternal chromosome. Maternal chromosome means mother's chromosome. Same, I am drawing one chromosome of the same type. This is paternal. Paternal means father's chromosome. So you can see that this is the arms of the maternal chromosome. I am shading one chromosome for you to understand. Okay, the shaded one is the maternal chromosome. The unshaded one is the paternal chromosome. So here you can see the two chromosomes. Imagine that this is the chromosome, one chromosome. Then one more pen I am using. So you can see that imagine that this is the paternal chromosome and this is the maternal chromosome. Okay, so these two chromosomes are actually crossing like this somewhere. Okay, that is what is shown here. So this point of contact between the two chromosomes is what is known as chiasma or chiasmata. So what is chiasma or chiasmata? How will you define this? The point of contact of the two chromatids of the maternal and paternal chromosome is known as chiasma or chiasmata. That means the mother's chromosome and the father's chromosomes are actually at a particular point they are having some sort of connection. There is a point of contact between the two chromosomes. This point of contact between the two chromosomes is what is known as chiasma or chiasmata. So this is actually what is called as crossing over. Okay, crossing over is taking place. So at this region what is actually happening? Mixing of genetic material will take place. So as a result of the mixing of genetic material, it will lead to variation. What is the meaning of variation? Variation means the differences seen among the offsprings. Okay? You know that your siblings are not alike. Differences seen in offsprings are called as variations. This is known as variation. If you are having a sister or a brother, you both will not look alike. There will be some changes. I am telling other than this twins. Okay, identical twins are there. They will look exactly alike. That is another thing that we will see later on. Other than that, identical twins, the appearance of the siblings will not be alike. It is because of variations. So variations are taking place during meiosis. Mixing of genetic material will be taking place due to crossing over. Okay, the term is like this, crossing over. So crossing over will lead to variations. Okay, so what is chiasma? The point of contact between the maternal chromosome and the paternal chromosome is what is known as chiasma or chiasmata. What is the importance of this particular point? At this point, actually mixing of genetic material is taking place, as a result of which variations can be found among the progenies. So that is the significance of meiosis. So mitosis, we cannot expect any variations. Now we will discuss what are the differences between mitosis and meiosis. Mitosis, I told you, it is actually a type of cell division that will be helping in growth and development. Whereas meiosis is the type of cell division that will be helping in the formation of gametes. Then mitosis, it maintains the same chromosome number. Meiosis, what is actually happening? The chromosome number is getting reduced to half in the gametes, so that during fertilization, the normal diploid number is reacquired. Then in mitosis, no mixing up of genetic material, so no variations. In meiosis, mixing up of genetic material is taking place, which will lead to variations among the offsprings or among the progeny. Then mitosis, only one type of nuclear division. Okay, only once the nucleus will divide. But meiosis, the nucleus will divide twice. Okay, we are not learning in detail about meiosis. Meiosis you will learn further in standard 11. So I am just giving a brief idea. So in mitosis, the nucleus will divide only once. Only one nuclear division will take place. But in meiosis, two nuclear divisions will happen. Then, how many daughter cells are produced in mitosis? Mitosis will result in the formation of 2 daughter cells. And what about meiosis? Meiosis will result in the formation of 4 daughter cells, because I told you 2 times nuclear division will happen. So 2 times nuclear division means it will result in the formation of how many daughter cells? 4 daughter cells. So all these are the differences between mitosis and meiosis. Let us recall. Cell division. What is cell division? The division of cell into 2 daughter cells with the same genetic material. Kinds of cell division. There are 2 kinds of cell division. Mitosis, that is for growth and repair of the body parts, and meiosis, that is for the production of gametes, that is sperms and egg. Mitosis. Where does mitosis happen? It occurs in the somatic cells. Just before the division of the cell, the nucleus prepares for this change and doubles the quantity of DNA. This is the interphase or the resting phase. Here, no change in chromosome is visible, but actually it is quite active in synthesizing the DNA. Mitosis. It is completed in 2 steps: karyokinesis and cytokinesis. Karyokinesis. The changes that is taking place within the nucleus, which finally divides the nucleus into 2. It occurs in 4 main phases: Prophase, Metaphase, Anaphase and Telo Phase. Prophase. Centrioles start moving apart and reach opposite poles. Chromosomes become distinct. Sister chromatids attach to each other at a small region called centromere. Spindle fibers appear between daughter centrioles, forming the achromatic spindle. Nuclear membrane and nucleolus disappear. Metaphase. Each chromosome gets attached to spindle by its centromere. Chromosomes line up in one plane at equator. Anaphase. Centromere attaching the 2 chromatids splits. The 2 sister chromatids of each chromosome separate and are drawn apart towards opposite poles as a result of the shortening of spindle fibers. A furrow starts in the cell membrane at the middle in animal cell. Telophase. Spindle fibers disappear. 2 sets of daughter chromosomes reach opposite poles. Chromatids thin out in the form of chromatin fibers. Nuclear membrane is formed. Nucleoli reappear. The cleavage furrow starts deepening in the animal cell. A cell plate is laid down in the cytoplasm at the equatorial plane in plant cell. Cytokinesis. The division of cytoplasm. The cleavage furrow deepens totally in animal cell and separates the 2 daughter cells. The cell plate grows from center to the periphery, resulting in 2 cells in the case of plants. What are the differences between mitosis in animals and plants? In animals, asters are formed. Cytokinesis by furrowing of cytoplasm. In plants, asters are not formed. Cytokinesis by cell plate formation. Meiosis. Reduction division producing gametes. The most significant aspect of meiosis is that the number of chromosomes in the sex cells is halved. This is essential because when the male and female gametes fuse during fertilization, the normal diploid number of chromosomes is reacquired. What is the significance of mitosis? It helps in the growth of the body, It helps in the repair of damaged and wounded tissues. It helps in the replacement of the old and dead cells. It maintains the same chromosome number in daughter cells. Significance of meiosis. Chromosome number is halved in gametes so that on fertilization, the normal diploid number is reacquired. It provides for the mixing up of genes. Thereby, genetic recombination will be taking place, as a result of which variations will be seen among the progeny. What are the differences between mitosis and meiosis? Mitosis. It takes place in the body cells. Meiosis. It takes place in the reproductive cells. Mitosis. 2 daughter cells are produced. Meiosis. 4 daughter cells are produced. Mitosis. Single nuclear division. Meiosis. 2 nuclear divisions. Mitosis. It helps in the growth and development of the body. It also helps in the replacement of worn out tissues. Meiosis. It helps in the production of gametes. Mitosis. It occurs continuously throughout life. Meiosis. It takes place only in reproductively active age. Mitosis. Full set of chromosome is passed on to each daughter cell. Meiosis. Only the half the number of chromosome is passed into the gamete. That is haploid number of chromosome. So that on fertilization, the normal diploid number is reacquired. Please do subscribe Bio Digest. Like and share. Thank you.